So in order to kind of catch up and keep us on schedule, I'm going to do the rest of Chapter 10 as a recorded lecture. If you have any questions about this lecture and the information here, please either email me or stop by my office or make an appointment and we can go over whatever topics you have questions about. Most of this is pretty straightforward information, so I don't think this will pose much of a problem for us. Within Chapter 10, we're talking a lot about marine ecology, and we should talk about the different types of lifestyles that marine organisms have. And these are things we've actually already addressed in other chapters. First, we have our benthic organisms. Those are the ones that live on the benthos, or they live on the bottom of the seafloor. They may be mobile, things like a crab, or they could be sessile, things like a sponge, where they're going to be attached and growing to the surface themselves. And then we have our pelagic organisms. The pelagic organisms are those that live in the water column. We have plankton, who can be either phytoplankton or zooplankton. They may not be able to swim very much, and they may not be able to swim at all, so they may be at the mercy of the current. This allows them to be carried from place to place, and we tend to call this type of lifestyle a drifting lifestyle. And then we can divide these up based on size, and we look at the different types of plankton, lifestyle, whether or not there are larvae in the plankton and then become something else as an adult, but we're not going to worry too much about that right now. The nekton are those that can swim enough to move past the current. So these are things that can swim against the current and move in the opposite direction of the current should they want to do so. While most of these things tend to be vertebrates, there are invertebrates that are capable of doing this, such as squids and octopus and other types of organisms. We can divide our major marine habitats by depth of water. First, we have the intertidal zone. This is the zone that is kind of adjacent to the land, as well as adjacent to the ocean. It is the land that is the area covered by water at high tide, but exposed land at low tide. So at low tide, it's an area exposed to air. At high tide, it's an area covered by the water. Then we have our subtidal zone. This is the part of the continental shelf that is underwater. So it's not going to be very deep. It's going to go out all the way to the shelf break. Our next is the bathyal zone. This zone is from the shelf break to 4,000 meters in depth. The abyssal zone is between 4,000 and 6,000 meters in depth, and the hadal zone is below 6,000 meters. We can look at these pelagic divisions as well as we look at what is in the water column. So these will be different than the last set because this is dealing with water column as opposed to dealing with bottom communities. So the Neritic is going to be our kind of inshore area. This is going to be above the shelf itself. So it's our near shore divisions. And then we have our oceanic pelagic ones. These are where you are offshore. You're beyond the shelf break. The epipelagic is from the surface to the first 100 to 200 meters. The mesopelagic is from 100 to 200 meters down to about 1,000. The bathypelagic goes from 1,000 to 4,000, and then the abyssopelagic goes from 4,000 to 6,000, and the hadopelagic is anything below 6,000 that is pelagic. There's not very much that's going to fall into that hadopelagic area. Whenever we talk about ecology, it's also important to talk about concepts like the flow of energy and the cycling of nutrients. These are two sets of processes that are really going to drive a lot of what we're talking about with ecology. And it's important to remember that energy flows through the system, so it enters and exits the system, whereas nutrients cycle through the system. Nutrients are always going to be recycled and reused, whereas energy comes in, is used, and leaves. When we talk about flow of energy, we're going to be talking about food. So you have autotrophs, heterotrophs, detritivores, and decomposers. Your autotrophs are going to be those that are your phytoplankton. These are your photosynthetic creatures or your bacteria that are chemiosynthetic, those that are capable of making their own food. Heterotrophs are those that are going to be eating food that others have made. They cannot create their own energy. Detritivores are those that are going to be consuming detritus, generally speaking, dead material, but they are separate from the decomposers, which are going to be the last step of what's breaking it down. Our decomposers are basically going to be bacteria, and archaea, where detritivores tend to be the animals and invertebrates and things that are eating the dead material. We can talk about trophic levels too as we talk about food webs. Your producers are going to be your autotrophs. These are the ones that are making the food into the system. Your consumers are your heterotrophs, those that are eating it. They can be a primary one, meaning that they eat the producers, a secondary one, meaning that they eat the primary consumers, a tertiary one, meaning that they eat the secondary consumers. And we frequently talk about a top consumer or a top predator. Or an apex predator. This is the one at the top of the food chain, the one who can eat everything down below. And food webs, you'll see on the next couple of slides what we're talking about with that. But it's important to note that not everything is going to feed on one level. Sometimes a secondary consumer is able to eat primary consumers, but also the producers. Within energy flow, our general rule of thumb is that 10% of the energy is passed from one trophic level to the next. And we can then assemble a pyramid of biomass, and we're going to look at each of these concepts very briefly. This is showing you, hopefully, as you can see, a food web. We have diatoms and phytoplankton as the base of it, separate from what you think of it on land because we don't have plants really, even though these are acting as our plants. We have the copepods, the amphipods, and the krill that in turn eat these organisms. So those are your primary consumers. Our secondary consumers are the small fishes and squids, the petrels, the Adelaide penguin, and the baleen whales. Our tertiary consumers, then our next level up, will be the large fishes, the skua, the killer whale, the emperor penguin could be up there, the Weddell seal, the leopard seal, and our killer whale is really our apex predator. He can eat just about anything in this system. But notice he's not going to focus on eating the krill. Those aren't going to give the food payoff that he needs. So he's going to eat the things that are going to give him the best type of biomass. This whole structure together is a food web. But if we want to talk about a food chain, that's what you see highlighted in blue. A food chain is just going to be step 1 to step 2 to step 3, looking at a specific line. So from the phytoplankton to the krill to the baleen whale. It's a very less complicated way of looking at it. And each part of the food web can be broken down into different types of food chains. And here we actually have that example broken down by levels. And notice in this aquatic system, we have a lot of secondary level or secondary consumers. In the ocean, this tends to be relatively true. There are a lot of things that eat on that secondary consumer level. There are also a lot of first level consumers or primary consumers, but they don't tend to have the diversity that you have on that secondary consumer level. And here is the idea of a food pyramid, but also that same food chain that we were looking at. And if you notice in this food pyramid, that rule of 10% does apply here. The phytoplankton can contain about 10 million calories, the krill about 1 million, and the whale about 100,000. So 10% as you move up of the energy is kept from layer to layer to add new biomass. Now, why might that be? Hopefully, you're thinking, well, what else are they doing with that food other than making biomass? And the answer is a lot of it is going to be used for metabolism purposes. And here we again have a food pyramid. Now it's good to know how much primary productivity there is in a system because that's going to tell you how much it can support of other life forms. And we usually use tracking primary productivity as a way of understanding what is kind of going on in the ocean habitat because primary productivity It's going to tell you how healthy an ecosystem is, what else is happening there, and really let you get an understanding of what's going on. Productivity is defined as the amount of carbon fixed in a square meter of sea surface per day or per year. We tend to measure primary productivity only with this, and that's going to kind of ignore quite a bit of the chemosynthetic, but that tends not to be in this area. Gross primary production is going to be the total amount of organic carbon that is made or that is fixed, whereas net primary production is going to be that gross minus respiration. So it's really going to talk about what is made through photosynthesis and respiration, but also the amount that is then available to the next trophic level. We will measure chlorophyll content sometimes, and that can be scaled up to primary productivity. Why do you think that is? Hopefully you're thinking, okay, chlorophyll is the green pigment in plants, in the chloroplast, that is needed to do photosynthesis. And if you know how much chlorophyll you have, then yeah, you can probably estimate how much photosynthesis is occurring. But productivity is going to vary pretty widely in marine environments, sometimes by season, sometimes by just the fact that things that are in the tropics are going to have relatively more productivity than things that are at the poles, at least on an average. But during a single season, that may not be true. We can estimate the amount of carbon dioxide that is taken up by our photosynthesizers by knowing how much chlorophyll there is, which can also allow us to estimate how much oxygen they're giving off. And when we do real studies of this, we frequently use isotopes in order to track this. And sometimes what we do to estimate our net primary production is we take 2 samples of water. We keep one in sunlight, we put one in the dark, and we see what the difference is based on the metabolism of the animals, because the ones in the dark are doing metabolism, the ones in the light are not. They're doing photosynthesis. And here we have kind of that estimating way of looking at how much primary production is occurring. We have 1 square meter of sea surface, and maybe we've put some sort of a plexiglass box or something like that around it. And we can measure then at the end what the concentrations of different things are in the water, like chlorophyll concentration, or like carbon dioxide or oxygen concentration, allowing us to see how much photosynthesis and how much respiration has occurred. This table gives you the typical rates of primary production in various marine environments. And notice that the Arctic Ocean does not fix nearly as much as what you find in the coastal temperate seas or in the upwelling areas, but the highest amount of fixed production is in the coral reef communities, which is probably similar to what you expected. The coral reefs, the salt marshes, the kelp beds, the tropical rainforests, all have very high levels of primary productivity. And notice that the tropical rainforests are listed here. That is to give you an idea of how productive the ocean is when compared to a rainforest. And surprisingly, seagrass beds, kelp beds, salt marshes, and coral reefs are as productive, if not more productive, than tropical rainforests at doing carbon fixation and primary production. So they're producing equivalent amounts of food available for their food webs. But what if you don't want to take water samples, or you can't take water samples? Because when you take a water sample, you're looking at just the primary production in that sample itself. What if you want to know what production looks like around the world? Well, we do have satellite imagery that is able to give us approximations of chlorophyll concentrations. Specifically chlorophyll A, which is the chlorophyll that you tend to think of when you think of chlorophyll. It's easily measurable with a lot of different lab tests, but it's something we can also measure with a satellite. And if we can take snapshots of chlorophyll concentration, it allows us to figure out about how productive the waters are. This doesn't always work over land for a lot of reasons, but over water, this is able to give us a great idea of how much chlorophyll is present. And as you look at this map here, what type of season do you think it was for those of us in the northern hemisphere? Take a look. What does the Arctic Ocean have as a color? Is it more green or is it more blue? Hopefully you're seeing that it's more green with some yellows and some reds, and that's indicating some high levels of chlorophyll concentration. And check out Antarctica, mostly blues. So if it has high levels of productivity in the Arctic Ocean and relatively low levels in Antarctica, that's hopefully going to clue you in that this is likely the Arctic summer or the Northern Hemisphere summer. So it would be summer for us in Philadelphia, while it will be winter for those of them in Brazil. Unlike energy, nutrients are going to cycle in our systems. They do not flow, they actually cycle. This means that we're going to have recycling of nutrients, which is important because it means that we will not run out of the raw materials required for life. Now, that may be a little weird to think about at first. I've heard it expressed in a couple really good ways sometimes. One of them is the idea that the water that we have on this earth has always been recycled. And the water that you are drinking today could be the very same water that Jesus drank when he was on the earth. That's not a disgusting way of looking at it. It's kind of a cool way of looking at it. The flip side of that is that the water that you're drinking today could have at one point in time been dinosaur pee. Less good way to look at it. So recycling is a good thing. It's a necessary thing, but it can be a little awkward to think about at times. Recycling is going to require decomposers, when we look at nutrients though, to break down dead material, called detritus, into water, carbon dioxide, nutrients, and other things. As they break it down and it becomes dissolved organic matter, or DOM. Organic matter because it still contains those carbon-containing compounds. And this is important because what it means is that when organisms die, while their energy is lost, their nutrients are not. The nutrients themselves are going to be recycled back. This keeps death from being the end of the nutrients. It brings them back into the cycle. So as the organism dies, the nutrients are recycled and made available for the next set of producers. And without decomposers, life would be horrible. It's not really an understatement. Without decomposers, we would have trash and dead things piling up everywhere because waste would not break down. Things will not rot without decomposers. That's very important to keep in mind. Decomposers are what allow that rotting process to happen. And that rotting process is kind of a not so great way to think about it. Instead, think about it as a process that regenerates nutrients, or that recovers nutrients, or that recycles nutrients. So rotting conveys some sort of a negative concept, but regeneration, recovery, recycling shows the importance and the positive side of what's happening here. We'll be looking briefly at the carbon, nitrogen, and phosphorus cycles, something that your ecology class is going to cover a little bit more in depth on probably. And it's important to note that in each of these systems, they originally came from either the Earth's atmosphere, the Earth's interior, or the weathering of rocks. And they're going through natural cycles. Human alteration of these cycles is occurring though, and that is causing some problems because we're making these cycles a little bit out of whack. Our first is the carbon cycle. The carbon cycle of the ocean is remarkably important. As you're all aware, carbon dioxide levels have been rising in the atmosphere. And there's been a lot of talk of maybe we can plant more trees or something to hold on to this carbon, to store this carbon. But the ocean holds 50 times the amount of carbon as the atmosphere. So if the levels are rising in the atmosphere, they're also rising in the ocean. That carbon in the ocean can be converted to biomass, and this happens through photosynthesis. It can be deposited as calcium carbonate through biogenic sediments. And this can be made then into the shells of our phytoplankton or zooplankton that are living in the ocean. It will then, as those die, sink to the bottom of the ocean, become biogenic sediments. Some of it will dissolve back into the ocean, but some of it will be then stored away. But the burning of fossil fuels is a big release, and this is where humans are altering the carbon cycle. And unfortunately, when we're seeing that productivity of the ocean is decreasing in some areas, what we're also finding is that the ability of the ocean then to hold that carbon or to serve as a carbon sink or carbon storage is also going to be decreasing. And one of the biggest concerns we have about the carbon cycle alterations is that the more carbon dioxide there is in the atmosphere and the more carbon dioxide dissolves into the ocean, the more acidic the ocean becomes, which actually decreases the amount of biogenic sediment stored as calcium carbonate shells because it increases dissolving. And it also makes life harder for a lot of the organisms that live in the ocean because it becomes a little bit more acidic, making it more dangerous to live there and more difficult to live there. Corals especially, we'll talk about this with the coral reef systems. And this is our basic carbon cycle. It's exactly what you would have figured. The atmosphere has some carbon dioxide, it dissolves into the water, it's used in photosynthesis, and it's produced through respiration. Very simple cycle. Nitrogen cycle is a little bit different. It is a very large and complex cycle. If you want to know a lot of the specifics of the nitrogen cycle, let me know. This is actually an area that I studied pretty in depth for my PhD. I'd love to spend some time talking with you about it. Truthfully, I could probably teach, well, a once a week course all semester, maybe even a whole course about this topic. So if you want to learn more about it, just let me know. Atmospheric nitrogen is pretty unavailable to those of us to those animals that live in the ocean. It's not going to be like carbon dioxide. It doesn't just dissolve in a form that is usable. While it will dissolve in perhaps as nitrogen gas, that's not usable for our organisms that live there. And the nitrogen cycle is going to involve gaseous phases, dissolved phases, and fixed phases. Gaseous means that it's in gas form. Dissolved means that it is in usually nitrogen oxide form or something like that, where it's in a chemical combination where it exists as dissolved in the system. Whereas fixed means that it is going to be in something that is alive, so then it's not available. Nitrogen fixers and lightning, surprisingly, make atmospheric nitrogen fixable or make it dissolvable into the ocean. The nitrogen fixers are things like bacteria or other things that will take nitrogen gas and make it into something else. Within the ocean, nitrate, which is NO3 negative, is perhaps the most important form of nitrogen that is talked about. And generally speaking, there used to be a balance between fixation, so what was brought into the ocean, and denitrification, what was lost from the system. And these used to be in a good balance. Today, we're discovering that that's not true. Human activities are producing and releasing a whole lot of fixed nitrogen, and that is altering the cycle. There is more nitrogen in the system that is dissolved or fixed than there should be. And it's made through fertilizers, through fossil fuel burning, animal waste, the cultivation of nitrogen-fixing crops, specifically soybeans. This means that we are altering the nitrogen cycle, and we're not sure in the long term what effect that will have. In the short term, nitrogen pollution is actually causing problems to a lot of ecosystems. It causes an overgrowth of epiphytes and of other types of organisms. It's linked to harmful algal blooms and a number of other topics we'll talk about this semester, as well as some that I talk about in environmental issues and some that are talked about in ecology. And this is what the nitrogen cycle is supposed to look like. But we have altered it quite a bit. I'm not going to test you on drawing these cycles. Please, please don't worry about that. The phosphorus cycle is a little bit different than the others. Phosphate is not coming from the atmosphere. The phosphorus cycle is going to be coming from rocks. Phosphate is the majority of the phosphorus that enters the ocean, and most of it's going to come from the land. It is picked up by water that weathers rocks, where water's breaking down rocks. It's picked up from bird guano, a nice way of saying bird poop. It's picked up from fertilizers that are on the ground surface. And very little of it is going to come in from the atmosphere. Chemical reactions have converted phosphorus sometimes into insoluble forms, which means that there may be phosphorus in the system that's not usable because it does not dissolve well. Different changes in pH and other things can make that happen. Phosphorus can be deposited in biogenic sediments or through chemical precipitation. And what is surprising is what makes it available to us again, then once it is locked away or stored away in these biogenic sediments, is uplift. When those sediments become uplifted to be part of a mountain or part of a coastline, they are then broken down again and dissolved away again by water. And if we were really going to spend a lot of time talking about nutrient cycles, we would have to talk about the silicon cycle and the iron cycle for the ocean, but we're going to kind of skip those. And there's our phosphorus cycle. We have two pretty basic concepts, I think basic concepts left. One is the idea of biodiversity. And biodiversity, as you know, deals with the variety of life on planet Earth. And biodiversity is unfortunately declining. This means we're seeing a loss of species throughout the planet. Extinctions have always occurred. Extinctions have always occurred naturally, though. And we're seeing today that the rate of extinctions is much higher than it otherwise should be. When we go through an extinction crisis or an extinction event, you're talking about extinction rates that are much, much higher than the background rate. And today we're seeing that these extinctions have a lot to do with us. Maybe it's pollution. Maybe it's habitat destruction. Maybe it's the idea of over or exploitation. The idea that we're hunting things and we're killing things at a level that they cannot sustain. Maybe it's climate change. Sometimes it's invasive species. But somehow species are disappearing. They're going extinct now faster than ever before. Faster than normal processes and faster than they should be going extinct. And we're left to wonder why. And I think we have to look at ourselves when we think about why. Conservation efforts are working, though. We have, for some species, been able to bring them back from the brink of extinction. And that is a good thing. And that is the place that you need to look for hope. Because the moment we say, here's an endangered species, and look, we have fixed this, we have turned things around, we've done something right. Unfortunately, they tend to focus only on the cute creatures. We call these the charismatic megafauna. The whales, the polar bears, the penguins, the things that people love. Because everybody wants to save them. But who wants to save the goblin shark? I mean, really. Maybe you do. Maybe others could. And sometimes it takes us talking about these organisms and helping people see that they are kind of cute and they are important to get them to care. Whales and sea turtles. We're helping them. We're bringing them back from the brink of extinction. Worms, seaweed, fungi, they're not really easy to get people to care about. But they are important for their ecosystems. They're important for food, for these other charismatic megafauna, and they're important for life on Earth. So hopefully we will see changes in the way that biodiversity is going extinct. Because when we look at biodiversity, we have to remember organisms do not live in isolation. They are part of food webs. They are part of nutrient cycles. Sometimes they're in a symbiotic relationship or a parasitic relationship or mutualistic relationship, and the loss of one is going to be devastating to the loss of the other. The loss of even the lowest species, or what seems to be the lowest or the least important species, is bad because sometimes it causes a cascade, a trophic cascade relationship that we don't understand because we don't know enough to predict what's going to happen when you lose one species. It's really, really hard to predict it when in the ocean we're just finally starting to understand how some of these systems work. We even know this is hard to predict on land what will happen if you lose one species. There's an idea, though, that we can save biodiversity, not by tugging on people's heartstrings, but by tugging on their consumer interest. The idea that things in the ocean might give us medications, or food, or other new raw materials that we need. Maybe also the idea of we don't know what's out there. Maybe something that's out there will be useful to us. For some people, that is what's going to get them. It's not going to be, oh, but the poor polar bear. It's going to be, yeah, but polar bears, you never know. Maybe we're going to find from them a way that we can develop a new diet aid or something. Who knows? That's something where it's going to get people to care. We do recognize, and hopefully you recognize, that biodiversity is important in and of itself. The fact that we have a great variety of life is good. Especially because it means that an ecosystem is healthier. From a Christian perspective, biodiversity is important. It is valuable. It is, as we see in Genesis, God talking about creation, good. Protecting biodiversity then is necessary from a Christian perspective because we're protecting the very things that God has created. There is quite a bit of fear from scientists about what the future will be like if we don't have very high biodiversity. In 1992, we set up an Earth Summit where they set up the Convention on Biodiversity. By 2002 at the World Summit, they found that we needed to significantly reduce the rate of biodiversity loss by 2010, and we didn't. At 2012 in Rio Plus 20, we agreed we had not made enough progress. We know that by using resources and energy more efficiently, we'll be able to actually cause less harm. We know that in order to be more resilient to disease and disaster, we need to have biodiversity, which means we need to stop with pollution, overfishing, and invasive species. Human life survives better with biodiversity because biodiversity provides us with oxygen, with food, with clean water. It breaks down our waste for us. It changes climate patterns. The loss of species, the loss of biodiversity, is going to be directly proportional to the loss of human health. So biodiversity matters. And there's good news. There are some problems, though, first. The problems are that our international agreements don't match. So if the United States wants to protect whales and Japan's allowing whaling, which is the fishing of whales, there's a problem. We need to have specific actions and goals and monitoring. This is like anything. If you want to get an A in organic chemistry, you set up a study schedule, you set up your plan for how you're going to do your homework, you set goals, and you check yourself along the way. This is the same way that this works in real life. We need to have actions, goals, and monitoring. Otherwise, it's going to be hard to get everybody on the same boat. The developed countries have to assist the poor countries. We can help them figure out how to do monitoring. We can help them reduce their pollutants. We can help them with sustainable development, as well as providing training and infrastructure and scientists to work with them. And we do need global monitoring programs through research, through technical assistance, and through the use of money well spent. The solution is pretty simple. We need strong international actions immediately. Like I said, simple, right? But maybe it could be. And we need to focus on management, protection, and research. Because if we don't, what happens next we might not like. And this is where the census of marine life comes in. And this is something that I find remarkably exciting. And I hope you do too. Prior to 2000, less than 1% of the ocean had been explored. Less than 1%. And they started a 10-year census of marine life. The idea was going to be to see if we could figure out what was living there, to have an idea of what lived in the ocean. Also, we wanted to know what used to live there. Perhaps it's gone extinct. And how can we then make predictions of what will happen with the loss of biodiversity? There were 540 expeditions that identified over 6,000 possible new species. So far, 1,200 formally described new species exist, and there are scientists still working on this. There were almost 3,000 scientists that worked on this at 700 different institutions from 81 different countries. So that last slide where I said international action was necessary and needed to be international action taken together, unified, it can be. We've already started doing this work. And we're working on making the information available. You can check out their website. They do have a place where scientists are able to put all of their data together so that we can use all of the data internationally to make decisions. It's hooked into the Ocean Biogeographic Information System, which means that you can provide information based on where organisms are, and those places can then be linked to all of the different data sets. There's also a goal to bring in historical data as well, because historical records have a lot of important information. It could be things like the genetic analysis of fish scales or whale bones. It could be old records that we have from fishermen about what we used to know about these ecosystems. And why does this matter? Well, you can use the Nova Scotian cod fishery as an explanation. We expected that it had been reduced in population by 95% by 1950s. Okay. Except we're wrong. The population had been decreased by 95% by 1850. So this goal of let's get it back to the levels it was in 1950 is really bad. We need to try and get the population to be healthy, to be back to the levels of the early 1900s, but probably 1800s would be good. But because we didn't know this information, we were actually able to figure it out using genetic technology and things like that. It was really helpful. So the Census of Marine Life is going to help us make better goals. What types of results are we looking at? Over 6,000 expected new species. So let that sink in for a moment. 6,000 new species. This has established a baseline for marine life. What's here? What used to be here? It's allowed us to map migration routes and breeding areas for many species, like the sooty shearwater tern, who makes a 4,000-mile migration. It lets us have an idea of what the impacts are on the deep sea from mineral extraction and from climate change, and allows us to see that those two processes are actually worse than the litter and the trash that we think of as being problematic, that we see as being problematic. It allows us to find that there's been a global decline in phytoplankton in fisheries. We knew there was, but this brings data to what had been anecdotal information. It showed us that the impacts of human species were actually worse than we expected. But we also found giant microbe mats, Jurassic shrimp, that we thought had been extinct for over 50 million years, the Loricifera group, which are 3 species that can live in areas with no dissolved oxygen at the bottom of the ocean, and they're multicellular animals at that. And we discovered that there are black smoker hydrothermal vents at areas closer to the poles than we ever expected they could be found before. So we found a mix of some bad in with all of the good. And then one of my favorite things we developed is a brand new tool called DNA barcoding. Because, well, we can't always get the data we want and bring it back to the lab and have everything alive. So what you do is you take a water sample and you analyze the DNA traces in the water, and you don't need to actually collect your organisms. It's a much safer way to gather information about what is there. And what you see here is kind of a system for that. Actually, what you see there isn't an arms, never mind. So the DNA barcoding is one thing we developed that's new. We've also developed new electronic satellite tracking tags, new systems for tracking movement of organisms. We've set up goals for management, and then this autonomous reef monitoring system is what's in the picture there. That is basically a reef condominium. So each arms, autonomous reef monitoring system is the arms, is a stack of 10 plastic plates left on the ocean bottom for a specified time period. Marine invertebrates sea squirts, bryozoans, sponges, decapods, snails, worms start living in it, much as like they would a coral system. After a year, or whatever the specified time period is, the scuba divers return, retrieve it, and they take it apart. The plates are photographed, the species are identified, recorded, sampled for DNA processing, and we've been able to use this information to figure out more about the tree of life. And some of these samples are then taken into the lab and studied so we can figure out how these organisms live. We've also developed a lot of information for the Encyclopedia of Life. So through the Census of Marine Life, we're understanding these ecosystems better than we ever thought possible. And we're realizing how much we still have to learn about the ocean, which for a lot of scientists helps us to embrace that idea of wonder and respect for these ecosystems. There are some very specific outcomes, though, from the Census of Marine Life. Scientists from all over the world worked together on this project, and they were able to build both personal and professional relationships. This is important. We have scientists crossing borders saying that working together matters. We have better understandings of how to sample and how to do new techniques that we never knew how to do before. And the scientists have been able to share knowledge so that what works in one ecosystem can be tried in another. And we can figure out how do we do science better. The scientists were able to create information that governments and managers could use immediately. Because oftentimes when we come to them with a scientific problem, governments and managers are saying, well, give me a solution or give me some data I can use. And this gave data, important data, in less than 10 years. It also showed us how much is left to learn. And it allowed us to really assess human impacts. But truthfully, for many of the scientists involved in this, it gave them a sense of purpose. And it helped them to embrace why they started studying the ocean in the first place.